Okay.
Good morning, everyone. Welcome to Orlando, Florida, to the American Heart Association meetings. I'm Dr. Brenth Muhlestein from Intermountain Healthcare, and I'm here with my good friend and colleague, Dr. Mark Effron, who is at the Ochsner Clinic and at the University of Queensland. He also has a previous career as being the medical director of Eli Lilly Company and is one of the world's experts in P2Y12 inhibition. And today they have a new study called the PharmClose study that talks about P2Y12 inhibition and pharmacogenomics. So tell us a little bit about what that study was.
So the PharmClose study was a study that was done in Italy by a group of investigators in which they looked at the genomics of clopidogrel metabolism. So there's a warning in the US label, and I think also in the European label, that suggests that patients who are CYP2C19 alleles, and basically a STAR2 allele, are non-responders, and depending on whether you have one allele or two alleles, and there's a loss of function, so you don't have adequate conversion of the prodrug into the active drug with clopidogrel. Most of it goes into an inactive metabolite if you have the STAR2 allele, especially if you have two loss of function alleles. And so what they did is they looked at patients and they randomized patients into standard care versus patients who had their care altered by whether or not they had the loss of functions allele. If they had the loss of function allele, they were then put on a more potent P2Y12, such as ticagrelor predominantly, or they might have had a couple of patients on prasugrel. And what they showed in their outcomes is that in their primary endpoint, which was a combined endpoint of ischemic events and bleeding events, that they did better in the genomic-guided therapy than they did with the standard of care. A very significant reduction, basically it's more than a 50%, almost 58% reduction in event rate.
And tell me what you think about that. Is that a real deal, or it seems sort of odd that you would get such a positive benefit, whereas the comparison head-to-head against clopidogrel didn't show that much of a difference if you gave everybody the more potent drugs?
So we need to look at it a little bit better and a little bit closer. The reduction is greater than any P2Y12 inhibitor has shown in any study, including clopidogrel versus placebo, prasugrel versus clopidogrel, or ticagrelor versus clopidogrel. So there's a much greater reduction in events. The reduction in ischemic events was also accompanied by a reduction in bleeding events, and that doesn't quite make sense because the reason you would expect to see reduction in events is better platelet inhibition. So you'd expect to see bleeding to either be the same or maybe slightly increased in the patients in the genomic-guided therapy as opposed to standard of care, and yet it was less. The other thing that is sort of out of sorts with some of the things that have gone previously is in the large pivotal trials of Triton TIMI 38 for prasugrel, in the Plato study for ticagrelor, that when these subgroups who were CYP219 poor loss of function alleles were evaluated, it had no effect on the outcome in the trial in those subgroups where they tested those genomic sub-studies. So now you have a study showing absolutely remarkable results, and those studies showing no difference. For what it's worth, there are also several posters here this morning at the American College of Cardiology meeting where they've also looked at genomic-guided therapy, and their results, one of the posters actually showed no difference to go along with the large trials. Another one showed a minor difference, but it was nowhere close to the difference seen in the PharmClose study. So I think we need more information before one can say genomic therapy. The other point that actually confuses me somewhat is the question comes in, is it the genomics or is it the functional response? And we know from prior studies done that about 10 to 20% of people who have even two loss of function alleles will still make adequate active metabolite of clopidogrel and have a good functional response, so that you do show platelet inhibition with it. Like it as well, patients who are homozygous for the wild type, actually about 20% of them show high on treatment platelet reactivity. So you can't say that just because I've got this allele or don't have the allele that functionally I'm going to have the same result.
Oh, that's all very interesting. One last question. So now prasugrel is generic and it's not inexpensive yet, but it's going to be inexpensive.
Hopefully.
Do you think pharmacogenomics for clopidogrel is past its time, because now we're going to have the better drugs that cost the same? Or do you think there's still some advantage for safety?
I think that I'm not sure the pharmacogenomics are going to lead to safety in any way, shape, or form, and I think that what we'll have to see is the patients who are getting it. In observational studies that have been done with Effient, which was the patented version of prasugrel, it's clear the physicians were basically choosing the drug based on safety, and so they would predominantly pick patients at lower risk of bleeding, not necessarily higher risk of ischemic events. What will happen when a generic form is out there, it's unclear. Will genomic testing really come in and play a role in the choice of P2Y12 inhibitor therapy? I think we need a lot more data before it should.
Oh, very good. It's always a pleasure to meet with you,
Thank
Dr.
you,
Effron.
Brent.
Thank you very much.
Thank you very much.
And thank you all for listening.
Okay, thank you.